Yeah. Good day, everyone. I'm Abiodun-Solanke Tomi, here to present the study titled Exploring the Potential of Orange, that is Citrus sinensis peel, as sustainable seafood preservative. I'm making this presentation at the second edition of the Biennial Conference of African Continental Association for Food Protection happening in Accra, Ghana. Seafood, as we know, is any form of sea life that humans consume as food. This can be fish or shellfish. Then it has a high demand in the food industry due to its high nutritional value. It's very rich in omega-3 fatty acid, which is an unsaturated fatty acid. Then because of its taste and texture as well. But despite these benefits that seafood has, it has been found to be highly perishable because of the high moisture content that it has. So, and there are several methods of preserving, that is extending the shelf life of seafood, and this method varies from canning, drying, salting, smoking. There are so many methods of preservation, but here in the developing country, Nigeria inclusive, smoking and drying are the most commonly used method. And this preservation of seafood is important, especially immediately after harvest, to maintain the quality and safety of the finished product. Before now, synthetic preservatives are commonly used, but these have health and environmental concerns attached with it. So the cross for this particular research is to put forward some natural preservatives that can be used in place as alternative to these synthetic ones. And orange peel is a natural is a waste product is a waste product that has shown potential to be utilized as a natural preservative. Though there are some other preservatives, but it has been found out that when you want to substitute synthetic preservative with a natural one that has competitive use, it may become more it will be very, very expensive, impacting negatively to the cost of the finished product. So the problem statements of this study are high post harvest losses, increasing the gap, that is widening the gap between supply and demand. Then we have also discovered, just like I told you, that smoking and drying are the most commonly used method, and many of the processes that are being used, even using the doing this smoking and drying, they are traditional methods of processes that are limited by poor quality fish, and the also longer duration of processing that impacts negatively on the quality of the final product you get from such processes. Then have told us that there is reduced interest in the use of inorganic preservatives due to the health and environmental concerns associated with it. Then many at times too, even this synthetic preservative, they're also very expensive. So the justifications are: one, with the growth in population and urbanization, that means that the future demand for good quality seafood is assured. Then oranges are consumed worldwide, and for example, in a place like Nigeria, orange is available all through the year, because as one orange from a region is going, another one is coming, so it makes the food products available, the fruits available almost throughout the year. And this waste is rich in bioactive compounds such as limonene, flavonoids, phenolic, and among others. And these compounds have antimicrobial and antioxidant properties, which suggest that they can be used as natural preservative for extending the shelf life of our food products. Appropriate smoking, spicing, and packaging have also been found to lengthen further the shelf life of food products. The objectives of this study are to investigate the possible use of orange peel as seafood preservative, to evaluate the safety of the fishes that are smoke dry using this natural preservative, and then to evaluate the effect of this preservative on the storage quality of hot smoked African catfish, that's Clarias gariepinus. So here we go to the materials and method. Here is a picture showing the different processes that are taking place during the smoke drying. So for the first one, the fresh averagely sized fish, the first thing that was done, sorry, let me just go to the slide, was the phytochemical and antimicrobial evaluation of the orange peel extract. This was done to investigate whether this particular plant extract will be good for what it is selected for. And then averagely sized fish were gutted, they were properly washed, and this was divided into two equal parts. As you can also see from this particular video, from this particular picture, some in a basket, some in a bowl. They were soaked into the orange peel orange peel with water, that is a solution of orange peel and water, while the other one was just prepared and processed without any preservative. So then the preparation of the preservative, the fresh oranges were bought, they were washed thoroughly, and these were peeled with the use of blade, so that we are sure that it's only the peel that we are getting, there is no pulp attached to it. And then the washed fish, the washed fish was soaked in the peel solution for an hour. This was allowed to drain, shaped, and then loaded into the smoking kiln. Then the fish was cooked and smoke dried until dryness was achieved. And this can be ascertained by doing a constant weighing of the fish products, so that as soon as you see that there is a constant weight, you know that the available water has been thoroughly removed. Then the fish was allowed to cool, it was packaged in appropriate material, and thereafter stored on the shelf at room temperature for 112 days. Sensory evaluation was done by a group of 30 panelists. Then samples of this the safety of this sample was done through microbial evaluation. And then as I told you, the fishes were stored for 112 days, and samples were taken every 14 days for the organoleptic and the microbiological evaluation. These are the tests that are recommended for shelf life evaluation. So here we go to the results. So from the results of the phytochemical and antimicrobial evaluation that was done on the orange peel extract, the results revealed that there was inhibition in the growth of some named mold, being a cause of the extract, that is the orange peel extract. Then the free radical scavenging assay for the orange peel aqueous extract against DPPH, that's diphenyl phenol, revealed that it has 71.2% activity, which I believe everyone here will agree with me that it's a very good percentage of activity. So this shows that this plant material selected for this study was found to possess good antimicrobial and antioxidant properties that can make them to be used as natural preservative, alternative to the synthetic one. So the drying time of catfish, so it was found that not only can orange peel be a very good um, replacement to synthetic preservative, it was also found that the orange peel treated samples dried faster than the control samples. So while the orange peel treated samples was thoroughly smoke dry at 8 hours, the control samples did not thoroughly dry until 12 hours. Then more moisture was also removed from the orange peel samples than the control ones, and then the average drying rate of the orange peel samples showed that 0.58 kg of moisture was being removed for an hour for the orange peel samples, while 0.45, that is a kg, as lesser than the orange peel samples, was dried. The was moisture was removed for an hour in the control samples. So here we see the picture, that is the graph showing the organoleptic evaluation of the orange peel and the control sample. So we can see here the red samples are the orange peel samples, while the blue samples are the control samples. So we can see that while the control samples reduced from 4/5 in the organoleptic assessment for the first day, the 14th day. That's the 14th day. We have 4/5 in overall acceptability, while the orange peel has 3.25. So the one that has 4, that's the control sample, reduced from 4 to 2.25 after 112 days of storage, while the orange peel sample that had a lesser organoleptic score in terms of acceptability, the 14th day, that is, it has 3.25, only reduced to 2.5. Then we can also see this even from the gradient, from the slope of the line, of the linear graph, linear line that was put on the storage samples. So while the one for control, the gradient was steeper than the one for orange peel. So this shows that of the two samples, even though the orange peel samples was rated low organoleptically for the first and 14th day, the control samples was rated higher, that is 4/5 compared to 3.25. Then but for shelf life, while the orange treated samples reduced from 3.25 to 2.5, that means that the organoleptic score of the orange sample was 2.5, higher than the one for the control samples that was rated higher initially with 4.00 to 2.25 after 112 days of storage. Then here we have the microbial table showing also the results, that is the volume of the microbial load of the two differently stored products, that is the control and the orange peel smoked catfish. So for the first day, the control have been having higher volume of microbial load control compared to the orange peel samples. The first day it has 2, while this one has none. 14 days 6, 2, and so on and so forth. So it can be observed from here that the orange treated sample had less volume of microbial good than the control samples. And as of the 70th day of storage, the control smoked catfish samples are too numerous to count number of that is volume of microbial load, which shows indicating spoilage, while the orange peel treated samples were still good even after 112 days of storage. The orange treated fish samples were found to reduce the growth of microbes to the barest minimum, despite not rated high in terms of the organoleptic properties. So conclusion: the natural compounds found in the orange peel extract exhibited significant inhibitory effect on the growth of pathogenic and spoilage bacteria in the smoked fish, not just bacteria alone, even for yeast and mold. These properties suggest the potential use of this extract as natural preservatives, enabling preservation with minimal health concerns. Then the orange peel slices has significant effect on the drying rate of the smoked product, aside the preservative ability that it has. So I'm hereby recommending that the use of orange peel is recommended for use as preservative and also as an antioxidant to extend the shelf life of smoked fish products. Then traditional methods of processing such as smoking, shredding, as I told you initially, in this part of the world, smoking and drying are the most commonly used method. And this, many of the players in this particular sector, they are rural women, they are artisanal fish processors that mainly stay in the coastal community, so that they can have they can be near the raw material. Discover that many of these people, is the traditional methods they have been using. One major difference in this particular study is the use of the natural, the traditional methods that they are used to. Because if it comes to a researcher, and many researchers now, what they do is they extract the active ingredients from this particular preservative. But many of these women have access to this extraction method and everything. So that's one of the major cross of this study, to see, using their own methods, what are those things that can be observed. And I was so glad when I saw that even without extracting the ingredient, the active ingredients in this preservative, they were still found to be potent as antimicrobial and antioxidant properties. They still have those properties that can make substitute the synthetic preservatives that have been used before now. Lastly, gaps resulting from this study. Then, yeah, so I know many of you may want to ask that, how come the sensory evaluation score given to the orange sample was lower than the control samples? So here I can see that one of the fallout of this particular study is that more researches should be done with varying concentration of the orange peel treatment, so that we are sure of the correct concentration to advise many of these local women to be using. You understand? So that we can see that we can make a proper balance between the organoleptic and then the microbial effects of this preservative. Thank you so much for listening.